Hello everyone, welcome back. Today I'm here with this part, and in this part I will discuss some questions from plant embryology, plant anatomy, and plant physio. Let's get started. So the first question is, bulliform cells are found in. Our options are maize, Gallisneria, Nerium, or Opuntia. So its right answer is maize. These bulliform cells are some specialized cells which are larger in size and are present in the upper epidermis of these monocot plants. So these large size cells are known as bulliform cells और ये present होते हैं upper epidermis में in some monocotyledonous plants, and their main role is in leaf rolling that we can see in this photograph. So this maize leaf rolling is done with the help of these cells and helps in prevention of water loss. The next we have this Nerium. Nerium जो plant है, it is again a xerophytic plant, and its leaf anatomy is unique. It shows multiple layers of epidermis, so multi-layered epidermis. So this is a unique feature of Nerium leaf. This Opuntia is succulent xerophyte where leaves are modified into spines, and the stem performs the function of photosynthesis, which is known as phyloclad. That is known as phyloclad. This Gallisneria is a hydrophyte and is a ribbon-shaped plant. The next question is, multi-layered epidermis occurs in. Our options are ficus leaf, Nerium leaves, Cycas leaflet, or both A and B. So I have already discussed this question. So the multi-layered epidermis is found in Nerium leaves. So this is the section of this Nerium leaf, and we can observe here. So there are multiple layers of epidermis, and it's then xerophytic adaptation. And this multi-layer epidermis protect this plant from desiccation or excessive water loss, and it contains these stomata, which are sunken stomata, which also helps in prevention of water loss. Then the Cycas leaflet, it is again it shows some specific tissue that is known as transfusion tissue, and it is unique to this gymnosperms. It helps in lateral conduction of water. Lateral conduction of water, because जो gymnosperms होते हैं, they lack the veins, or in place of these veins they bear this tissue which is known as transfusion tissue. The next question is, Erymerus is an example of having which type of endosperm? So our options are halobial endosperm, cellular type of endosperm, nuclear type of endosperm, or ruminant endosperm. So its right answer is halobial endosperm. I will discuss about all these type of endosperms. So we have the three different types of endosperm according to Sitchinarf 1929 classification on the basis of mode of development. First is the nuclear endosperm, second is cellular, third is halobial type of endosperm. So the first is nuclear endosperm. Here this primary endosperm nucleus divides many times, and its division is not followed by wall formation, so it leads to this multinuclear condition, and it is found in the plants like we have Limnanthus or Oxyspora. In some plants, nuclear endosperm becomes cellular at later stages, means there is formation of cell wall. Then this next type of endosperm is the cellular endosperm. As the name indicates here, this division of primary endosperm nucleus is further followed by immediate wall formation that results in formation of a cellular structure, and it is common in dicot. And in this type of endosperm there is a presence of this structure which is known as haustoria. So it is a structure which helps in absorption of nutrients. Then the third one is halobial endosperm. It is an intermediate form between these two type of endosperm, that is cellular or nuclear type of endosperm. So it is most common in monocots. And Davis observed the presence of halobial endosperm in 14 monocot families, and this development of halobial type of endosperm is found in again Erymerus. So this was the answer of this question. So this Erymerus is an example of having halobial. Next question is, pollination takes place through the agency of insects is known as. Our options are anemophily, zoophily, entomophily, or clistogamy. So its right answer is entomophily. So pollination by insect is known as entomophily. Anemophily is pollination by wind with the agency of wind. Zoophily means pollination with the help of animals. And clistogamy, these are the flowers which are closed flowers, and here cell pollination occurs. जो flowers enclosed होते हैं, and these flowers are known as clistogamous flowers. So pollination is the act of transfer of pollen grain from the anthers of one plant to the stigmatic surface of the another flower of the same plant or of the different plant of same species. So this process is known as pollination. So this transfer requires various agents for transfer of pollen grains, which may be abiotic agents or biotic agents. Abiotic agents include air and water, while biotic agents include various insects, animals. So pollination is basically of two types. One is self-pollination, that is called autogamy. Then we have cross-pollination, that is also known as xenogamy or allogamy. For cross-pollination, it requires different agencies like abiotic agencies, which include wind and water. So for these we use the term anemophily for wind pollinating flowers and hydrophily for water pollinating flowers. The next, biotic agencies. So it required different animals or insects for transfer of pollen grains. Firstly, we have this cantherophily. Here this pollination occurs with the help of beetles. So transfer of pollen grains occur with the help of beetles, that is known as cantherophily. The next is phelinophily. Phelinophily means pollination with the help of moths. Then melitophily, that is pollination with the help of bees. Then this is psychophily, that is pollination with the help of butterflies. Then melycophily, pollination with the help of snails. Ornithophily, that is pollination by the birds. Then chiropterophily, that is pollination with the help of bat. And the last one is the myrmecophily, that is pollination with the help of ants. Then next we have this next category, that is called anthrophily. So this is pollination with the help of humans, that is called anthrophily. The next question is, closure of stomata in normal plants can be induced by, our options are, this question is from plant physiology part, that is from hormonal part. So our options are indole 3 acetic acid. The next is abscisic acid or GA, that is gibberellic acid, or benzyl aminopyrin, which is an example of a synthetic cytokinin. So its right answer is ABA. ABA is commonly known as water stress hormone. तो जो water stress hormone, that is ABA, और ये help करता है to prevent the water loss by closing stomata. So this is the main function of this abscisic acid. It also brings dormancy in plants or seed or birds. So dormancy को promote करता है ABA. Basically this is responsible for opening and closing of stomata. It acts with these calcium ions, and these calcium ions along with other ions like potassium ion affects the opening and closing of stomata. We have different plant hormones and they have their unique functions in the plant. First of all, we have abscisic acid. It maintains the seed dormancy and winter dormancy and helps in closing of stomata. Next is auxin, which is commonly known as master hormone, and it is derived from a Greek word auxine, which means to grow. So it promotes stem elongation. Main role is in apical dominance. Then it also helps in root formation, in stem cuttings, root initiating hormone, then fruit growth. Inhibit axillary bud outgrowth means it helps in apical growth, that is apical dominance, and prevent the later bud growth. And it also prevents leaf abscission. Next, brassinosteroids. So these promote stem and pollen tube elongation, promote vascular tissue differentiation. It is again performed by this IAA. So this IAA also helps in vascular differentiation. Next, cytokinins. These are mainly involved in cell division. This hormone is mainly involved in cell division. It inhibits leaf senescence. So if we put a leaf on the cytokinin solution, so it will delay senescence, and that process is known as, that phenomenon is known as Richmond-Lang effect. Richmond-Lang effect. So this effect is shown by this chemical which is known as cytokinin. It also affects root growth. In tissue culture, there are two hormones whose concentration defines the callus differentiation into root and shoot. So cytokinin to auxin ratio decides the root and shoot formation. So more is the cytokinin, lesser is the auxin, it will lead to the formation of shoot. And if it is reversed, means lesser is the cytokinin and more is the auxin, so there will be formation of roots. So this is how these two hormones plays important role in tissue differentiation in tissue culture. And next is ethylene. This is the only gaseous hormone. It promotes fruit ripening and leaf abscission. It inhibits stem elongation and gravitopism. In addition to this, the unique feature of this ethylene, it shows triple response. That is shortening of the coleoptile or stem toughening, then horizontal growth, and increase in the diameter of the settling. So these are the responses which are shown by ethylene. The next hormone is gibberellin GA. It is obtained from one fungus that is called Gibberella fusariae. It promotes seed germination, means it prevents seed dormancy or break seed dormancy. It can also substitute vernalization, that is cold treatment which is required for flowering in some plants. And it promotes stem growth, internodal growth, and fruit development. Break winter dormancy, that is can substitute vernalization. Mobilize nutrient reserves in grass seeds, so it also helps in seed germination in the monocot seeds. So these are some important features of this hormone. In addition to this, gibberellin also helps in this phenomenon that is known as bolting. So bolting is the stem elongation in some rosette plants. Rosette plant means the plants which have highly reduced stem. So ऐसे plants में ये stem elongation करता है और flower produce करता है, that is called bolting. Like we have this cauliflower, radish. So in these plants it helps in this phenomenon that is known as bolting, that is stem elongation and flowering. So this was all about for today's discussion about these questions. Hope you'll get some idea from this presentation. Thanks for watching. Have a nice time.